Dear students, Assalamu alaikum. Today we are going to talk about our course Molecular Biology, course code BCHM 221. This is lecture number 6 of the series of our online lectures. And the topic of our today's lecture is transcription in prokaryotes, but we will mainly focus on the process of initiation of transcription in prokaryotes. So this is why I have given this initiation in the bracket for you to understand that although the overall topic is transcription in prokaryotes, but we will focus on the process of initiation mainly, and then like in the process of replication we studied initiation separately, and then we talked about elongation and termination. So similarly here we will first talk about initiation of transcription in prokaryotes, and then we will talk about the processes of elongation and termination. So this is because this is again a big topic, so this is why it is divided into 2 lectures for you to be able to understand it easily. So the outline of today's lecture is we will talk about what is transcription. It means we'll talk about the definition and the related information with transcription. We will talk about RNA polymerase and the enzyme which is mainly responsible, or which has a very important role in the overall process of transcription. So we will talk about the enzyme RNA polymerase. Then we will talk about phases of transcription, like the process of replication. Transcription also have 3 distinct phases. One of those phases is initiation, which will be the focus of our today's lecture. Then of course there are processes of elongation as well as termination, but we will talk about the processes of elongation of transcription and the process of termination of transcription in the next lecture. So for today we only mainly focus on process of initiation of transcription. So moving to the topic now, the first thing that comes to mind is what is transcription. So transcription is the process by which the information in a strand of DNA is copied into a new molecule of messenger RNA or mRNA. So basically this is the let me remind you that in the central dogma of molecular biology, we know that information from DNA is copied from to mRNA and then to the to protein. So this is a kind of phase in which it is the process by which the information in a strand of DNA is copied into a new molecule of messenger RNA. So this is overall the process of transcription. Now let me also elaborate that DNA is, safely and stably stores genetic material in the nuclei of cells as a reference or template. But this information in DNA, which is safely and stably stored, has to be transcribed first before it is translated into protein. So this process of transcription, in a way, work is an intermediate between the process of between DNA and protein. So meanwhile, messenger RNA is comparable to a copy from a reference book, because it carries the same information as DNA, but is not used for long-term storage and can freely exit the nucleus. You know that we say that DNA work is storage of information, but DNA remains in the nucleus. So how can this information be carried out? So this information is carried out through the process of transcription, where messenger RNA is formed, and this messenger RNA has the capability to move freely, to freely exit the nucleus. This is why, because the process of transcription it occurs in the nucleus, while the process of translation occurs in the cytosol. So this information from messenger RNA, it freely exit the nucleus, and once it exit the nucleus, it is able to provide coding for the process of translation. Now, to elaborate it further, although the messenger RNA contains the same information, it is not an identical copy of the DNA segment, because its sequence is complementary to the DNA template. So it shows that the information is not identical to the copy of DNA segment from which it is transcribed. Rather, it is its sequence is complementary to the DNA template. So this is very important information that you should know. The sequence should be complementary. Now, transcription is carried out by an enzyme called RNA polymerase. RNA polymerase is not the only factor. There are a number of accessory proteins called transcription factors. These transcription factors, they also play an important role in the process of transcription, but the main enzyme is RNA polymerase. So now, what is RNA polymerase? We briefly try to understand what this RNA polymerase is. RNA polymerase in E. coli have molecular weight of around 450,000. It has 4,158 residues. It consists of these number of residues of amino acids, and it consists of 5 polypeptide chains. The number of polypeptide chains in RNA polymerase are 5. These chain overall consist. These chains overall make the enzyme RNA polymerase. There is a difference between DNA polymerase and RNA polymerase. The difference is that, unlike DNA polymerase, RNA polymerase does not require a primer to initiate synthesis. Here, I would like you to recall the process of DNA replication. In DNA replication, the DNA polymerase always required a primer. It was provided by the primase enzyme, and in case of lagging strand, it was provided in the form of primer again in the form of Okazaki fragments. But the beauty of RNA polymerase is that it does not require a primer to initiate synthesis. Now, initiation of transcription occurs when RNA polymerase binds its specific DNA sequences called promoters. Now, the importance of promoters is very obvious, because DNA is a macromolecule, and if a certain segment of DNA has to be transcribed, The RNA polymerase should be able to bind close to that particular segment of DNA which is to be transcribed. Now, if RNA polymerase binds randomly to any part of DNA polymerase, this will result. This will be a hugely wasteful process, because RNA polymerase will have to transcribe a huge region, and then the region of interest will be very small, and it will have to be a wasteful process. So this is why in reality the process of transcription occurs very close to the region which has to be transcribed, and this is RNA polymerase is helped by a region called promoter. So RNA polymerase binds at specific DNA sequences that are called promoters. So we talk about this. I'll show you in the diagram how the promoter is located. But before this, in prokaryotic organism, transcription occurs in 3 phases known as initiation, elongation, and termination. The process of initiation, elongation, termination, you have seen this in the DNA replication. In DNA replication, we also have processes of elongation, initiation, elongation, and termination. So the process of transcription also occur in 3 phases: initiation, elongation, and termination. RNA is synthesized by a single RNA polymerase enzyme which contains multiple polypeptide subunits, 5 to be precise, as I told you before. The 5 subunits, RNA polymerase consists of these 5 subunits. Now, in E. coli, the RNA polymerase has 5 subunits. This form of the enzyme, in which it has all the 5 subunits, is called holoenzyme. The sigma subunit may dissociate from the other subunits to leave a form known as the core enzyme. So the sigma subunit of RNA polymerase has the capability to associate with the RNA polymerase or dissociate with the RNA polymerase. So in situations it can associate, in other situations it can dissociate from the RNA polymerase. So when the sigma subunit, when it dissociates from the other subunits, it leaves a form known as core enzyme. When all the 5 subunits are there, it is the RNA polymerase in the form of holoenzyme. Now, initiation of transcription. Transcription, as I told you, cannot start randomly, but must begin specifically at the start of a gene. Gene, you know, the segment of DNA which codes for a certain protein. So transcription must begin specifically at the start of a gene. Now, signals for the initiation of transcription occur in the promoter. Sorry about this. In the promoter sequence, occur in the promoter sequence, which lies directly upstream of the transcribed sequence of the gene. The promoter contains specific DNA sequences that act as points of attachment for RNA polymerase. So these are the promoter region function is point of attachment for RNA polymerase. In E. coli, two sequence elements are recognized by the RNA polymerase known as -10 sequence and the -35 sequence. So they are present there. -10 means that 10 bases upstream of start of transcription, and -35 means 35 bases upstream of the start of transcription. So the exact sequence, the exact promoter sequence can vary between promoters, but all conform to an overall pattern known as the consensus sequence. So there can possibly be small differences. They can vary, but overall pattern is known as consensus sequence. The sigma subunit of the RNA polymerase is responsible for recognizing and binding the promoter probably at the -35 box. In the absence of the sigma subunit, the enzyme can still bind to a DNA, but binding is more random. So this is it is showing the importance of the sigma subunit, that it helps to avoid random binding of RNA polymerase to DNA. The enzyme RNA polymerase covers about 60 base pairs of the promoter, including the -10 and -35 boxes. So here is the diagrammatic representation of the process of transcription. Here is the -10 region, and here is the -35 region. Now, the process of initiation consists of transcription consists of generally two phases. One of those phases is binding, and the other one is initiation. Here is the RNA polymerase, and here is the sigma subunit of the RNA polymerase. This +1 is showing the start point of the process of transcription, or downstream from here it is represented by plus sign, and upstream from the start point it is represented by minus. So this -10 region means that it is 10 base pairs upstream, and -35 is showing that it is 35 base pair upstream. So in the binding, phase, the initial interaction of RNA polymerase with the promoter, this is the promoter region, with the promoter leads to formation of a closed complex. So RNA polymerase, it binds with the DNA in the promoter region, and it forms this closed complex. Now once this closed complex is formed, the process of initiation of transcription begins. In the closed complex, here the RNA polymerase is stably, the DNA is stably bound but is unbound. But here in initiation, one can see here a 12 to 15 base pair region of DNA from within the -10 region to position +2 or +3 is then unbound to form open complex. Additional intermediates, additional proteins, they are not shown here, have also been detected in the pathways leading to closed and open complexes, along with several changes in protein conformation. The initiation phase overall encompasses transcription initiation and promoter clearance. Once the elongation complex the sigma subunit is released and polymerase leaves the promoter and becomes committed to elongation of RNA. So from here the phase of elongation of transcription begins, which we will talk about in the next lecture. So hope you understand now the process of initiation of transcription. If you have any question, I would be very happy to answer. Thanks for watching.